Hello everyone, I'm Yuanyuan from some scholarship and I did my PhD and some scholarship but now I'm a postdoc from PMC. Today, I mainly talk about my work from my PhD study, the search for life biosignatures on Mars, the detection of DNA with TMAH thermochemolysis on SAM. And we know Mars has been very popular these days, and we are trying to search for life biosignatures on Mars. And this is because the past environmental conditions on Mars are like the Earth today. And this means the Mars could be conducive to the emergence of prebiotic chemistry or even the origin of life. Until now on Mars, we have found the perennial liquid water, some organic molecules, and there are energy sources. So if it's possible to find some life biosignatures on Mars. And to answer these questions, we need to do the in situ analysis by rovers. We had Viking, Curiosity, Perseverance, and ExtraMars. And we also have the lab simulation experiments. And the lab results will support the data collected by this rover. And among these rovers, we had Viking, Curiosity rover, and ExtraMars. These three rovers, they have their sample analysis instruments, and by SAM and MOMA, and they can do the in situ analysis of the Martian samples. And during the in situ analysis of Martian samples, we will apply the derivation method. Here we can see on SAM, we have different capsulars containing derivation reagents. Here, the green colors, the TMAH and MTBS, TFA, and DMF. And they already have done some experiments with the derivation method. For example, in last year, November, we did the first TMAH experiments on Mars, and today we mainly talk about the TMAH thermochemolysis. So what's the TMAH thermochemolysis? It's like we have non-volatile compounds, but this means it's very difficult to be detected by our device. But if we apply our TMAH thermochemolysis, the non-volatile compounds will become volatile and it can be detected by our device. Here we give one example. When we apply our TMAH to analyze fatty acids, the methyl functional group from TMAH will replace the active hydrogen of fatty acids. So at last, we will have the fatty acids derivatives. So during the TMAH thermochemolysis, it will help to increase the volatility and decrease the polarity of the sample. So we can say in this process, the TMAH thermochemolysis played a very important role. And as we mentioned before, we are trying to search for life biosignatures on Mars. And we know there are so many important organic compounds that are important for life, but for DNA and RNA, they are essential for life because they carry the information of life. So we'd like to say if it's possible to detect the the DNA from life cells with or TMAH thermochemolysis that have been applied on SAM. So to achieve these goals, we started with a very simple chemical standards. First, we apply our TMAH thermochemolysis to analyze the 7 nucleobases, the nucleosides, and the nucleotides. And then we applied polymers. Here it's poly A because this is the RNA tile. And next we applied the TMAH thermochemolysis to analyze the DNA fragments from the live cells. Here we choose E. coli is because E. coli is very common in lab and very easier to get it. So all these experiments, they have been done with the pyrolysis and gas chromatography and mass spectrometry. This device has the same mechanism with same instrument, so we can apply the PYGCMS to analyze, to do the simulation experiments in lab. First, we did the nucleobases with TMAH thermochemolysis experiments. We only take the adenine as an example. So when we apply TMAH to analyze the adenine, we can say the methyl functional group from TMAH will replace the active hydrogen from adenine. So we have different derivatized adenine. From the result, we can say we have different peaks of methylated adenine. This is because for adenine, it has isomers, and when we apply TMAH to the derivation, we have different cases. We have one methyl replacement and two methyl products, and the three methyl functional groups replacement. So that's why we have different peaks from TMAH thermochemolysis with adenine. And we also compare the peak intensity of adenine at different temperature. We can say with the increase of temperature, the intensity of methylated adenine is highest at 600 degrees. So we say that 600 degrees is the optimal temperature for the detection of nucleobases. And next, we analyze the DAMP, and DATP with TMAH thermochemolysis. When we can say from the result, we found their typical structure, the methylfurfuryl methylated phosphate, and we found the methylated nucleobases are the main products from these DNA fragments. And surprisingly, we also found the methylated DA from DAMP, and DATP with TMAH thermochemolysis. And this means our TMAH can protect the structure of our DNA fragments. And when we analyze the nucleobases, we found that temperature can influence the detection of DNA fragments. So we optimize the temperature for the detection of DAMP, and DATP. We found 200 degrees is the optimal temperature for the detection of these DNA fragments. And we found that if you'd like to detect the phosphate, the temperature should be at 300 degrees. And to detect the deoxyribose, the temperature should be at 600 degrees. And for the detection of nucleobases here, the temperature should be at 200 degrees. Here, this is the structure of poly A, and we can imagine if we analyze the poly A at very high temperature, this poly A could be decomposed to several parts. But when we apply our TMAH to analyze the poly A, we found their typical structure of DNA fragments, and we also found the methylated DA from poly A with TMAH thermochemolysis. So again, this proves that TMAH can protect all poly A from degrading. And last, we analyzed the DNA fragments from the E. coli cells, and we can say from the result we found almost all of the nuclear bases with our TMAH from our E. coli cells, and also we found the typical structure of like the methylphosphate, methylfurfuryl. And this means our TMAH thermochemolysis can be applied to analyze the DNA fragments from cells. So, and last, we conclude that our TMAH thermochemolysis can be used to search for live biosignatures such as DNA or RNA fragments from live cells, but this is only the first step for the detection of live biomarkers from cells. And next, we can analyze the bacteria that have been detected in mushroom analogs. That will be more interesting, and our results will provide important reference data for the interpretation of data collected by the rovers. And last, I'd like to thank for the support of French Space Agency and SAM MOMA funding, and also thanks for the support of CSC scholarship. This is all my presentation. I'd like to take any questions. Thank you.